Oh, hey, biology people. Just doing some light reading. All right, let's get started. Today we're going to talk a little bit about evolution and evidence support for Charles Darwin's theory of evolution. And we're talking about scientific support, not emotional support. All right, so today we're going to be talking about fossil record. We're going to go over comparative anatomy, vestigial structures or organs, comparative embryology, and last but not least, molecular biology. And by the way, this pointing is pretty tricky because it's actually over here and do backwards. Up still up and down still down, but left and right are backwards. All right. So this is about how we know um, that Darwin's theory of evolution due to natural selection is correct. Remember, he came up with this idea, wrote in a book about 150 years ago, and today it's stronger than ever. So the question is, in that past 150 years, as we've learned more and more and more, it just really kind of confirms his theory of evolution through natural selection. That's what we're going to talk about today. So we're going to talk a little bit about the earliest way we kind of were able to put together pieces of the evolutionary puzzle, so to speak, was through the fossil record. So let's start off by saying, what is a fossil? Fossils are the preserved remains of ancient organisms. And remember, it's not a complete record because mostly, A, it's really hard to become a fossil, and B, um, all soft body organisms don't make very good fossils. Not a lot of jellyfish fossils out there. But the fossil record does provide evidence about what's been here before. We find it in sedimentary rock, and the fossil record is predictive as well, meaning you can say, here's an organism 400 million years ago with front feet and a fin in the rear. And you could say, I bet if I look at organisms in rocks 350 million years old, I might see a similar organism with front feet and instead of maybe a tail, two nubs in the back. And so you can make predictions and then go find it in the fossil record as well. When it comes to looking at the fossil record, we start seeing ancient organisms, and we're talking, organisms as young as 10,000 years ago to organisms that are hundreds of millions of years old. So when we talk about the fossil record, like how do we know how old a fossil is or from which area it came from? There's two types of kind of dating as far as telling out how old something is. Relative dating, and then what we're going to talk about in the next slide or two will be radioactive dating. But relative dating is determining a method of a fossil's age by comparing it with other knowns in the rock layer. And it's kind of more of a general term. So if you're literally hiking down the Grand Canyon and there's bands that you can see in the Grand Canyon, relative dating would say the fossils that you see at the top are going to be younger than the fossils that you would find at the bottom. And in some cases, fossils at the top tend to be more complex than fossils at the bottom. Not always, but you would think that that would happen as we go kind of farther down in the evolutionary record. So again, this is a picture, it almost looks like the cross section of the Grand Canyon. I would encourage, side note, if you ever get an opportunity to go to the Grand Canyon, you can literally walk through history. You can see seashells in the middle of Arizona as you descend down deeper and deeper into the Grand Canyon. And again, relative dating would say when you're at the bottom, those are the older fossils compared to the ones at the top. Now, that gives some good general ideas about how old something is. But if you want to get very specific, we can use something called radioactive dating, which is how we can calculate a more precise age of the fossils based on the amount of certain radioactive isotopes that it contains. And this is very similar, kind of the analogy of cutting down a tree and looking at the rings and counting the rings. You can easily say, okay, this tree is 88 years old because it puts a layer every year. So radioactive isotopes are based off of a half-life. It's kind of like almost like a stopwatch that these isotopes decay at a constant rate. And depending on the element you look at, you have different half-lives. So some elements have a half-life of, in the weeks or the months or the minutes. Some have a half-life that's in the billions, and some have a half-life that's in the thousands. So a common one that we talk about a lot, we talk about half-life. And again, it's the length of time required for half of radioactive atoms in a sample to decay, is carbon-14. And we see this a lot with man-made objects. If you've ever gone to a museum and seen, maybe a Native American clay pot, and it'll say that this is from, this pot is 3,500 years old. It's not because they lifted it up and there's a made on date on the bottom. They're like, oh, that's when it was made. They're able to take carbon-14, which has a half-life of 5,730 years, and see what's left in there and just kind of count backwards to see how old that piece of antiquities is. So a lot of man-made objects, they do carbon dating with carbon-14, and then with older objects, they might pick a different element. So to give you an example, if you start with 100 grams of carbon-14, you only have 50 grams left after 5,730 years. So based off of that, again, scientists are able to kind of calculate pretty precisely how old certain fossils are using radioactive dating. All right, so we talked a little bit about the fossil record, and we talked a little bit about relative dating and carbon dating, and now we're going to get into comparative anatomy. And we're talking about comparing animals in a species and seeing the similarities they have. Comparing parts of organisms for similarities in structure and function to see if the organisms have common ancestors. So again, for example, all mammals share a lot of similarities. I like this picture. This is a pretty cool picture. Right here we have a picture of, again, this pointing is pretty hard. On the way left here we have a human hand, and in my human arm here we have a humerus. We have in the forearm we have the radius and ulna, two bones here. We have carpals, our wrists, and then we have our fingers and our phalanges. If you look at the cat structure, what we have here is again, we're seeing the same structure with a slightly different function. So my arm and a cat's forearm is very different as far as function, but you see the same pattern of a humerus, a radius and ulna, carpals, which are highlighted in yellow, and then modified phalanges. Again, we have here a whale's fin. Whale's a mammal, and we see the same structures again repeated. And so you see in a whale's fin, you see a humerus, a radius and ulna, carpals, and then phalanges. And again, we have the same structure, different function. My arm and a whale's fin, very different function. So these are examples of homologous structures, and we see this across all mammals. So if you look at a bat's wing, is very similar to a human arm as far as structure, obviously not function, and it does vary quite differently from that of a bird. Sorry, you don't see the whole picture because my face is in there. But it gives us an idea of common ancestry and how natural selection works by not starting over every time, but making slight mutations and modifications to an existing structure to form eventually a different function that nature says makes that animal or that organism or that trait more fit to survive in the natural environment. An analogous structure, these are just kind of some terms. We talked about homologous, homo being same structure. An analogous structure is a structure that are made quite differently but have the same function. You see this a lot in animals that live in the same environment, right? So a good example of analogous structure, a bird's wing causes the animal to fly, a bat's wing causes the animal to fly. Very different structure. A bee's wing or a moth's wing, again, causes the animal to fly, completely different structure. Those would be analogous structures. My favorite thing. Again, we're talking about support for evolution, and what do we see, whether it's in the fossil record or we see evidence in comparative anatomy, how similar organisms are who are closely related. We have this fun thing here called vestigial structures or vestigial organs, and these are structures that were functional in our ancestor, but that are no longer really useful in the body today. They're almost like leftover historical markers of things how we used to be. A good example, kind of a classic example, is again right above me here. This is an example of this is an Atlantic right whale, and if you look at an Atlantic right whale and you were to cut it open, you shouldn't. There's only like 400 left. They're very endangered. But if you were to cut it open, one of the things you would find in this Atlantic right whale right above my head is that they have a pelvic bone just floating in their body that's not really attached to anything. And so the question is, what is a whale doing with a pelvic bone? This is a good example of the vestigial structure. Again, we take this information here and we pair it with the fossil record. All organisms on Earth have pelvic bones because those are where legs are attached to. It helps it for animals that walk on land. And we look at a modern whale today and we look at the fossil record, it shows us how whales evolved actually from land animals. A vestigial structure in modern humans, you would see over to my boom there, is our wisdom teeth. Again, a lot of you guys are in high school, and right about the age of like 15 or 16 or 17, you get 4 extra teeth moving into your skull. Now, 500,000 years ago, if we were walking, in sub-Saharan Africa, and you actually made it to 15, way to go, that's pretty tough. But around that age, it would have been an advantage to have 4 extra teeth come in about that time. Probably would have lost 1 or 2 in a fight, maybe a hip-tooth tiger ripped out, who knows. But it would have been an advantage. Today, thanks to modern dentistry, they're more of a vestigial structure because they're no longer needed. Kind of something that we used to need, but we don't need anymore. And we have some vestigial responses in our body too. When we get cold, we tend to get goosebumps on our skin. It raises the hair on our body. Now, it doesn't really do much now. You can be like, oh, your skin looks weird. But, again, 600,000 years ago, when humans or hominid species had much more hair over their body, raising the hair on the body would have kept warm air against the skin and helped us out. All right, vestigial organs. So we talked about vestigial organs. We talked about comparative anatomy. And then we have here a picture of 2 guys with a box of fossils. I actually don't know who those 2 guys are. All right, now we're going to talk a little bit about comparative embryology. And we're talking about comparing embryos and organisms of similar species evolving over time and showing the similarities in the embryonic development of species. Now, it makes sense that animals that share a lot of DNA are very similar in their development. And to keep that in mind, 96% of our DNA is the same as that as a pig. Okay, so we're not that special. Here we go. So comparing the formation and development of embryos across different species. So I have a couple different species here. Everything from a human right above me, right there, human, to a rabbit, to a cow, to a pig. Now we start getting, those are all mammals. Then we go into the next one, chicken, birds, tortoises, reptiles, salamanders, amphibians, and then we have a fish. And as the farther away we get evolutionarily speaking, we start to see a lot of differences. But animals in the same, class or group here, in this case mammals, you see that we have a lot of things in common. And at one point, it's nice to know that at one point you and I had tails and gill slits. So what this shows, though, it kind of also shows a little bit about molecular biology, which we'll talk about in a second. But it shows that again, organisms of closely related species have similar development in utero. If I show you this picture, well, I'll talk about this first. Early stages of development in many vertebrates are very similar. It shows common ancestry. Human embryos still have gills and a tail. We talked about that. They have the same. They develop tissues and organ layers at the same process and the same speed in many cases. Here's a picture of a human embryo, and you can see here at 21 days. If you look at the one at 21 days, and I don't have a picture, I apologize, but if you look at a bottlenose dolphin at 21 days, you couldn't discern the difference. They almost look almost completely identical, which is pretty cool. Now, molecular biology. Again, Darwin had no idea about DNA. He didn't really understand the whole genetic process. At the same time that he was writing his book, Gregor Mendel was doing his work with pea plants. But molecular biology is the study of genes of organisms to see how important similarities you see in the DNA level. DNA similarities, can show evolutionary relationships, can show how closely we are related to certain organisms. The greater similarities in DNA, usually the closer we are related to that species. this is an example of kind of DNA samples run out on a gel, and you can compare, how they compare like the crime scene DNA to suspects. And what they do is when they run out these gels, you can start to see certain fragments. Well, when we start looking at modern DNA, and we look at amino acids of certain proteins, when I might say to you, you are 94% identical to a pig, what I'm saying is if we took out all of your DNA and we took out my DNA, we took out a pig's DNA and we looked at the actual codes of proteins and structures, 96% of that DNA is identical. All right. That's what's meant by that. And we can see certain similarities. You can pick a certain hemoglobin, and the difference between a hemoglobin protein in a human and a hemoglobin protein in a rhesus monkey might be just like 1 or 2 amino acid difference. So we can see these similarities quite a bit. So it's a lot of information as far as molecular biology, but it kind of leads to the question, when we talk about evolution, who are we related to? Genetically speaking, here's a picture of a chimpanzee, but not to burst the chimps bubble. I think our closest molecular DNA relative is actually an orangutan. The orange one from the Jungle Book. Which also, I feel like we need to talk a little bit about when it comes to evolution. A lot of people get upset when they see like a picture of above here, and they hear the word evolution. They see a person and a monkey, and they say, maybe they went to the zoo and they saw, a monkey like throwing his poop, and they're like, I just can't believe I evolved from this animal. That's not what evolution's saying. Evolution's not saying that you evolved from this chimpanzee, or you or I evolved from an orangutan. I think the analogy I've used in class before, that'd be like parking a Toyota next to a Toyota Prius next to like a giant like monster truck and being like a Prius evolved from the monster truck. That's not what happened. But that Prius and that monster truck have a common ancestor in the Model T. Just like you and this chimpanzee here, you didn't evolve from the chimpanzee, and the chimpanzee didn't evolve from you. But you'd probably go back in human development and chimpanzee development. We go back hundreds of thousands of years, or 5 to 6 million years, that there's a common ancestor where something kind of diverged off. So that's what we're saying. All right, we're going to talk a little bit in the next PowerPoint about what makes a species a species. How do you go from species A to species B? All right, I'll talk to you guys later.